Good afternoon. My name is David Tichy. I'm an attending physician here at the Children's Hospital of Philadelphia, and I'm here with Megan Martin. And today we're hoping to talk to patients and families about clinical trials and clinical research currently ongoing at CHOP. The objectives today are kind of going to give a brief overview of how clinical trials have impacted our ability to cure childhood cancer, then go through some of the different types of clinical trials and clinical research, learn about some of the processes that are involved in participating in clinical trials, discuss some of the resources that are available for patients and families at CHOP, and then very briefly go through some of the ongoing clinical trials that are open right now at CHOP and some of the breakthroughs that are happening in our center every day. I think it's important to highlight how much clinical trials and clinical research have impacted cures in childhood cancer. And I think this is something that's recognized all over the globe as far as how much it's impacted and improve the way that we can cure children. Survival in childhood cancer continues to improve, and it really continues to improve because of the success in clinical trials. 80 years ago, if a child was diagnosed with cancer or an adult was diagnosed with cancer, they were universally fatal diseases. And nowadays, if you look at all children who are diagnosed with cancer, over 85% or the vast majority are cured. We still have a ways to go because our ultimate goal is to cure every child who has cancer and eventually cure every adult who has cancer as well. I think to understand how clinical trials have impacted cure rates in childhood cancer, sometimes it takes helpful to do a history lesson. The most common childhood cancer is acute lymphoblastic leukemia, and I'm going to kind of tell this as a story of how we got to be where we are today based on some of the breakthroughs that happened in this one form of childhood cancer, and similar things impacted other childhood cancers as well. I think it's important to note that the first breakthroughs in any cancer, either adults or children, was actually in childhood leukemia, and that was because of clinical research. And I'm going to take you back to the 1940s. So in the 1940s, children who were diagnosed with leukemia uniformly did not survive their disease. And there was a physician, a pathologist, so somebody who looks under the microscope primarily instead of treating patients, named Sidney Farber. And in World War II, he was studying soldiers who became anemic. So their red blood cells got low, they got really tired. And it was noted then that it was because they had vitamin deficiencies. And that was the first time that vitamins like folic acid and B12 were used to treat anemia or vitamin deficiencies in patients. When he looked under the microscope, the cells, the red blood cells and white blood cells in somebody who had a vitamin deficiency looked identical in his mind to children who had a certain type of leukemia called acute lymphoblastic leukemia. So he surmised if vitamins could make the soldiers who had vitamin deficiencies better, maybe these same vitamins could help children with leukemia. So he treated a few children with leukemia with vitamins, and not surprisingly, it did not work. But then he had the opposite question. Well, maybe he actually thought the vitamins might have made them worse. So if the vitamins might have made them worse, maybe if we did the opposite, if we block vitamins, if we block the way that they're chewed up or made or broken down in the cell, maybe that would be effective in leukemia. So we turned to a scientist, a biochemist, Dr. Rao, who used to be at Harvard with him and who moved into pharmacy, into industry, and he asked, do you have any drugs that could actually block vitamins? And he was actually working on a number of drugs that were used to block vitamins, and one of them was called aminopterin. And this was the first chemotherapeutic medicine, the first medicine really used to treat cancer. Dr. Farber did a clinical trial. It was a small clinical trial at his center. It treated 16 children, and 10 of them with aminopterin went into remission, which means that you couldn't see the leukemia cells under a microscope anymore. This was the first time with any type of cancer that patients were actually put into remission where the disease was helped. All of the children, unfortunately, the leukemia came back except for one child. That child was named Jimmy, which if you've ever heard the Jimmy Fund on TV, it was named after him. He probably actually didn't have leukemia. He probably had the monovirus, which is probably why he got a durable remission and cure with that. But this was the beginning of clinical trials and making breakthroughs to help childhood cancer. One thing that was also important back then was it couldn't just be done with doctors and scientists alone. They had a great idea, they had a new drug, but what they didn't have was the ability to try it in more patients, to move into bigger clinical trials, and that required resources and funding. And that actually was helped by a lady named Mary Lasker, who was a tireless advocate for childhood cancer research back in the 40s, 50s, and 60s, eventually helped form what became the NCI through advocacy in Capitol Hill, Other patients and families who had lost children, including the DeVillers, who formed the Leukemia Lymphoma Society, started forming foundations to help donate to childhood cancer research. And it was actually a partnership between science and medicine and philanthropy and advocacy that actually really spearheaded the beginning of clinical trials in childhood cancer research. Based on that work, a number of doctors and scientists made a number of breakthroughs over the next 10 to 20 years, starting with Gertrude Elion, who was a biochemist in New York who invented a lot of the drugs that we use today. Most of the agents that we use to treat cancer in 2020 were actually developed in the 1950s, 60s, and 70s and actually came from nature. Don Pinkel and Hansard Ring were two people who came up with the idea of, instead of treating children with one drug and getting them to remission, the concept of using multiple drugs together over sequential blocks of time. And really, these are the people that actually had the first cures. So basically, it went from an uncurable disease, ALL, to a disease where about 60% of children were cured back in the 1960s, early 1970s. And the therapy they used then is very similar to what we use now. And they did this through clinical trials and clinical research. A big difference back then from today was Don Pinkel was at St. Jude, Hansard Ring was in Germany. At both of these places, because the cure rates were so low, you could actually do a clinical trial, perform it from start to finish at a single center. But once the cure rates got better, you needed to treat a lot more children to move the goalposts and to improve cures. Which brings us to some of the things that are roadblocks and make it a little bit more difficult to do trials and actually treat children with cancer. One is it's rare. Cancer is common in adults. 1.6 million adults will get cancer in the next year. In contrast, if you add up all forms of childhood cancer together, about 10,000 children in the US, less than age 15 years, will get cancer. That makes it so it makes it more difficult to perform clinical trials or do clinical research where you can test new drugs because you need to have a number of patients to test it on to see if you improve the cure rates. Childhood cancer can impact anyone. It affects all races, it affects all ages, it affects all ethnicities. And I think it's important when we do clinical trials and clinical research that we can't just study one population. We need to really know, do our drugs and our therapies improve cure in different populations, different ethnicities, and different races as well. Because childhood cancer is rare, years ago, a number of clinicians, doctors, nurses, scientists formed what's called cooperative groups. And many of the trials that are open at CHOP are what we call cooperative group trials. So if you're an adult and you get diagnosed with breast cancer, for example, you could go to one of 20 different hospitals in the US and get treated quite differently. With childhood cancer, because of cooperative groups that have been around for over 50 years, you were diagnosed with leukemia or a brain tumor, and you're treated at CHOP, or you're treated at Harvard, or you're treated in California, or you're treated in Alaska. The therapy that we use is nearly identical because we all kind of partnered together to treat children the same way and to form these cooperative groups to do clinical trials. So regardless of where you are, you're treated the same and you have access to the same kind of trials. This is just showing a map of all the different places in North America and then a few other countries that participate in the Children's Oncology Group, which is what runs a lot of the clinical trials that are currently open at the Children's Hospital of Philadelphia for children who are newly diagnosed with cancer and some children that relapse. If you just look at North America, pretty much all over the US, there are centers that participate in the Children's Oncology Group. And then drilling down on our area, you can see there are a number of places in just our area, including CHOP, St. Chris, for example, DuPont, that are members of COG, so they participate in the same clinical trial network. Even though we now cure 85% of children, we still have a long ways to go. Each year, if you look at age 0 to 19, well, it said 10,000 for less than 15. It's about 15,000 for when you get to be children, young adults, and adolescents. Approximately 1 in 285 children will be diagnosed with cancer before their 20th birthday. If you look globally, there are more than 300,000 children diagnosed with cancer each year. And if you look globally, and at the US in particular, about 20% of children with cancer will not survive it. So we need to do better at curing more kids. Every 3 minutes, a family hears the devastating words that their child has been diagnosed with cancer. And cancer remains the #1 cause of death by a disease for children in America. And the only way that we're going to move the goalpost to curing everybody is through clinical research and continuing clinical trials. So one of the main areas in childhood cancer that we're very interested in is bringing new drugs and new products and new therapies to the clinic. And it's a very complicated process. And I'm going to kind of go through in a couple slides the process of why does it take so long? Why, when you hear about this new wonder drug, does it take 10 years sometimes before they can actually get access it for my child? It's a lengthy process. It usually takes about 10 to 20 years from a new drug being identified in the lab to being FDA approved. It takes even longer to get approval in children. That'll be something that Megan will talk about in a little bit, about the ethics of doing clinical trials and research in kids. And it usually costs about $2 billion for any new drug to actually get it FDA approved. There's a number of steps in the drug development process, from discovery to laboratory research to clinical trials to the government reviewing it to post-marketing, which I'll go through in a second. You've probably seen a lot of this actually in the news lately of the way that we're getting COVID vaccines to patients, going quickly in a much more hurried time pace, getting something into patients and approved within a year versus our typical timeline of 10 to 20 years. And it's important to highlight that there's doctors and scientists and nurses and others that are involved at all process of the steps at CHOP. So the first step is discovery. So this is basically what would be a better way to treat cancer, to cure cancer. People studying in the lab, how do cancer cells cause disease? How do they escape our natural body's mechanisms to prevent cancer cells from arising? Every day we are exposed to toxins and chemicals and other things that lead to a single cell in our body getting changed so it forms a cancer cell. But normally our immune system, which is a part of our body that fights infections, goes in there and wipes out the cancer cells. When you get cancer, why does your body fail to do that? We need to understand better how cancer cells are resistant to some of our current treatments. And then we need to develop new agents, and that includes what people think about as new drugs, pills, things you get by IV, but also in modern medicine, gene therapy and cell therapy, which we'll get to at the end. The next step of the process, which usually takes a few years, is actually testing your new idea, if you have a new drug or a new compound, in the lab. Before you want to expose adults or children with cancer to something new, you really want to make sure that it works in a petri dish, to test it in mouse models, usually mice, to make sure that it doesn't have unexpected side effects and it actually works in those preclinical models. Just to highlight, there are a number, they're listed here, it's too many to go through, of clinical and basic science research labs that are at CHOP that are studying new drugs as a new way to bring them to the clinic. And there are countless examples of things that we've tested in the lab at CHOP that are now in clinical trials in patients, and some of them are now FDA approved and have become standard of care. The third step is actually clinical research. That is the clinical trials, which we'll dedicate the rest of the talk discussing. For any new drug to get FDA approved, which means that a doctor can just write a prescription, they have to undergo many clinical trials, first in adults and then in kids. And Megan will go through the different types of trials there are so you can understand the different steps in the process. After clinical trials are completed, which can take a number of years, the FDA then reviews all the data. And then they want to make sure it's effective, it's safe. They don't want to just review the clinical trial data. They want to review all the stuff that happened in the lab beforehand. And then within 6 to 8 months, sometimes a year, they decide if they're going to approve the drug or not. And then they give it what's called a label. A label is the official approved use for a drug. A lot of what we do in medicine, both in cancer medicine and outside of cancer medicine, is what people call off-label, which means the drug is we have a new drug that's approved to treat diabetes. That drug may also work to help childhood cancer. If we use it in children with cancer, it's not an approved use. You can still prescribe it and sometimes work, but it's not per its label. And I think it's important to understand that distinction because if something is labeled, the insurance companies pretty much have to pay for it. When you do things off-label, they have the ability to refuse to pay for it a lot of times, which is why sometimes if your child is in the clinic and it takes a long time for them to get this new drug they want to treat it with, sometimes it's us fighting with the insurance company. Because a lot of what we do in childhood cancer and in children in general is what we call off-label use. So we're using an agent not for the way that it was approved originally by the FDA. And then after that, for years, with any new agent, data is collected at different centers and sites and sent to the FDA to make sure there aren't side effects, aren't things that were not seen in the initial clinical trials because they're very rare. With initial clinical trials, you may try something in 1,000 people to see if the drug works and is safe. But there are some side effects that only affect 1 in 100,000 people. So after the drug's approved, sometimes you don't see that until you start treating patients around the country, not as part of a trial, but as part of standard of care. With that, I'm going to pass it on to Megan, who's going to kind of talk about the different types and phases of clinical trials.
Thank you. So the main goal of phase 1 clinical trials is to test to see if the drug is safe, to identify any side effects, and to find the best dose. Children are not little adults. They metabolize drugs differently, so finding the best dose is especially important. These studies typically involve 10 to 50 people, and they can be done in healthy volunteers or people with a disease or condition that's being studied. If a new drug is for cancer, researchers would conduct trials in patients with cancer. Most drugs are tested in adults first and then children, and about half or 1 in 2 drugs will move on to the next phase of trials. So the goal of phase 2 clinical trials is to find out if a drug may help treat a disease. These are relatively small studies, so we can't definitively say if it helps. It would be a really high bar to do so. They typically involve 20 to 300 people, and the study drug is given to patients that have the disease that's being studied. They also provide us with more data on side effects, and less than 1 in 3 drugs will move on to the next phase. The goal of phase 3 clinical trials is to prove if the drug helps patients. These studies typically involve hundreds to a few thousand people, and they can take 3 to 7 years to complete. Often enrollment into these studies is usually randomized, which means that whether or not a patient gets study treatment is determined by chance or the flip of a coin. And these studies also help us to identify different types of side effects, like those that might be more common or rare or more long-term. And less than 1 in 4 drugs tested in phase 3 clinical trials moves on to the next phase. So the last phase is called phase 4 clinical trials. This is also known as post-marketing surveillance, which Dr. Tichy just reviewed as part of the drug development process. And these occur after the drug or device has been approved. So there are several different types of clinical research. There's correlative research or biology studies in which we collect samples from a patient in order to study disease. These could be saliva samples or tissue samples or blood samples, depending on the type of research that's being conducted. Population studies follow large groups of people. So these studies could look at a group of individuals who share a common characteristic, or they may involve looking at a large group of people to review their response to a drug or their risk of getting a disease. And then lastly, as we just discussed, clinical trials, there's observational trials and interventional trials. Observational studies document the course of disease. They usually look at 2 different populations that differ by 1 characteristic. And then interventional trials could look at a new drug or therapy. They could study different types of therapies, or they could be supportive care in which there's no study drug being used, but the goal is to improve the health or quality of life. So there's a relatively new piece of legislation that went into effect in August of last year. This is called the Research to Accelerate Cures and Equity Act. And its goal is to increase the number of studies devoted to pediatric cancer therapeutic trials. So in the last 20 years, only a handful of cancer treatments were developed and approved specifically for children. But now with the RACE Act, drug companies are required to test possible new therapies in children as well as adults. The ultimate goal is to improve children's cancer treatments. There are exceptions, however. If there is no childhood cancer that the drug could help, then they would be exempt from this new law. And the legislation does only apply to cancer, not other types of therapeutic areas. So before enrolling onto a research study, a patient must provide consent. This is a process, not just collecting a signature on the document. And the consent document that we review with the patient and the discussion with the study physician and patient's family should always contain the following information: the purpose of the research, the number of patients that will be participating, a description of study procedures, any medicines or tests that the patients will be required to take, risks of the study drug and also possible risks of any study procedures, possible benefits to participating, any alternatives to participating in the research, and then any costs that the patient might incur while they're participating in the research. So in addition to all of that information, we also want to make sure that we include the statement that the trial could end at any time by the study team or by the drug company, We're constantly updating trials as we learn new information throughout the different phases of clinical trials. There's also information on who to contact if the patient has any questions about the research. And then we always want to emphasize when we're having these discussions with the patient that participation is voluntary. There's no penalty for saying no. They won't lose any access to care or benefits, and patients can always stop participating in research at any time. So a family meeting is a family-centered meeting that happens with the patient and their family and study physician. It's basically a discussion surrounding the plan of care. It is included in the patient's medical record, and it provides a really nice documentation of the consent process and all of the elements that we just discussed. So patients have to undergo screening tests in order to determine whether they're eligible to participate in a study. The screening tests may occur before or after the consent is signed. And basically, we want to make sure that it's safe for a patient to participate and that the patient has a type of cancer that's likely to benefit from it. These screening tests can include blood tests, electrocardiograms, echocardiograms, x-rays, CT, MRIs, etc. And sometimes they can even include biopsies of your cancer. Each clinical trial has a protocol that the study doctor and team must follow, and the protocol outlines a list of eligibility criteria to help determine, one, if you have the type of cancer that might potentially benefit from participating in the study, and two, to determine if it's safe for you to participate. Some of the eligibility criteria is very broad, and sometimes it's very specific. For example, a broad eligibility criteria would be any child with leukemia, but something more specific would be children between the ages of 3 to 10 with the third relapse of a rare genetic type of leukemia isolated to the brain. So in addition to the results of your screening tests, your study physician is also looking at your medical record, and they help determine whether or not it's safe and whether or not you're eligible to participate in the study. For early phase studies, so for phase 1 and phase 2 clinical trials, a lot of times the number of slots or openings for that study is really limited just because those studies are so small to begin with. So the study team and the study doctor are always checking in to see what's available to patients at any given time. Some studies involve what's called randomization. We talked about this a few slides ago. And this basically just means that whether or not a patient receives study drug is determined by chance, like flipping of a coin. And then there's also multi-stage research in which sometimes 2 consents need to be signed. So for example, for some of our CAR-T or immunotherapy studies or bone marrow transplant studies, a patient would sign 1 consent to collect the cells and then a second consent to receive treatment with the cells. Similarly, with our leukemia trials, there's 1 consent for the first month of treatment, and then the patient signs a second consent for the rest of the treatment. In cases like these, it can involve 2 separate family meetings and consent discussions. So we want to make sure that patients that participate in clinical research are protected. And there are 2 founding documents that help ensure this. The first is the Declaration of Helsinki, which is widely known as the cornerstone document on human research ethics. It was originally established in 1964 in Finland, and it's basically a moral code for physicians involved in research and ethics. It actually begins by stating, it's the mission of the doctor to safeguard the health of the people. His knowledge and conscience are dedicated to the fulfillment of this mission. Now, the second document that I wanted to mention is the Belmont Report. So this was written based on discussions held in 1976, and it basically establishes 3 principles and guidelines for the conduct of research in human subjects. The first is respect for persons, which means that people can make their own choices. The second is beneficence, which means that we do no harm. So we want to maximize possible benefit and minimize harm. The third is justice. All persons are created equally, so we want to make sure that we have a selection of research subjects. The second bullet point here, I'm sure you're all familiar with, Food and Drug Administration. That's the federal agency that's responsible for protecting and promoting public health through the control of food safety, prescription and over-the-counter medications, vaccines, and medical devices. There are also local, state, and federal laws in place to help ensure patients are protected. CHOP also has several entities here at our institution to help oversee the protection of human subjects in research. We have a regulatory board, which I'll talk about next, and then we also have an office of compliance. So federal law requires that an independent review of any research that involves human subjects takes place. And this independent review committee is called an Institutional Review Board, or IRB. And the goal of the IRB is to review, monitor, and approve human subjects research. CHOP IRB committee membership includes faculty from a wide variety of clinical areas and specialties, legal advisors, nurses, pharmacists, community members, non-scientists, and individuals not affiliated with the institute. So I just wanted to pivot a little bit and talk about the Center for Childhood Cancer Research here at CHOP. Our mission is to eradicate the pain and suffering of childhood cancer through a highly integrated program of basic translational and clinical research built on an outstanding clinical care program. According to U.S. News and World Report, we're the #1 pediatric cancer program in the United States, and our program is led by Dr. Stephen Hunger, who's the director of the Cancer Center, as well as the chief of oncology here, and Dr. Tichy, who is the director of clinical. Our center is involved in research areas including cell therapy and transplant, immunotherapy, neuro-oncology, hematologic malignancies, neuroblastoma, survivorship, solid tumors, developmental therapeutics, thyroid cancer, behavioral oncology, and cancer. And I also wanted to mention that we have 2 new programs that we've opened over the last year, a very rare solid tumors program and our vascular malformations program. We're excited to be able to offer patients research opportunities in these areas as well. We currently have over 200 studies open and enrolling patients. These include clinical trials and observational biology studies. Back in December of 2020, the CHOP Research Institute created what's called the Research Discovery Finder. This is basically a tool to help patients and families find open studies to participate in at CHOP. You can search for studies based on conditions or study phase and study location. And what's really nice is that each entry has a link to all of the investigators' profiles and their departments, so you can see what their research, what they're interested in research. So some of our newer studies within the Cancer Center are listed on the Research Discovery Finder, but not all of our 200 plus open studies are listed. And the reason is that anytime we want to put any sort of information on a public website, we have to get certain approvals in place. So the IRB is required to review and approve all of that public information. And because many of our studies were open before the Research Discovery Finder was developed just 7 or 8 months ago, we're still in the process of getting all of those approvals in place to have all of our cancer studies, Cancer Center studies listed on that website. But that's definitely a goal of ours, and we're hoping to have that ready and available within the next couple of months. If you have any questions regarding clinical research, you can always email us. That email address that's listed on the slide is monitored several times a day, Monday through Friday. And you can always find additional information on research topics on our Cancer Center. I'm going to turn it back over to Dr. Tichy.
I think it's important to highlight that some of the things like the online messaging is reviewed by groups like our IRB, because the last thing you want to do is have any center like CHOP or somewhere else that provides patients or families with misleading information. So they want to make sure that we don't have something that says this new drug cures everybody. We want it to be fair, balanced, so patients really know what they are coming to get, because we don't want somebody to come to CHOP, especially if they live far away, and then find out that the way we discuss something or describe something online is not really as promising as it seems to be, which is why they go through a lot of levels. The last thing I'd like to just talk about very briefly are some of the active and ongoing clinical trials and clinical research that's ongoing at CHOP, and just giving a couple examples. I could actually fill another hour just talking about this if I went through all the different types. Historically, in the treatment of childhood and adult cancer, the way that we treated cancer was with kind of what we call non-specific medicines that just attack things that grow fast. Cancer cells grow fast. They grow faster than most other parts of our body. Most of our chemotherapy medicines that have been around for the past 50 or 60 years, they work by just blocking or destroying things that are growing really fast, so they don't hurt or cause any problems with healthy parts of our body. So while they may make you do things like lose your hair, because that grows fast, or affect your blood cells because those grow fast, those things are replaceable. Most drugs don't do things like damage your heart or your lungs or your brain, which are harder to replace and don't grow fast. In the late 90s until now, we kind of started thinking about treating cancer differently, which was, instead of having sledgehammers that just attack any cancer cell that's growing fast, try to understand how cancer works and then develop what we call more targeted therapies. So specific drugs that are designed only to work on certain types of cancers based on the genetics of them, the biology of them, what makes the cancer cell tick, what's specific about a certain type, that you have a drug that's not going to work broadly in lots of cancer, but may work very specifically and be very effective in rare types of cancer or certain types of cancer, almost more what we call a personalized medicine approach. So if in the old days, when somebody was diagnosed with leukemia, everybody got it treated the same way. And for the most part, if you're newly diagnosed with leukemia, you're going to get treated the same way no matter who you are or where you are. However, if it comes back or you have certain types of leukemia, you may get therapy that's more tailored to you by doing complicated tests on the cancer cells in labs at CHOP to see if there's something that's different that would make you respond to medicines differently or not respond. One example of this is a type of leukemia, type of acute lymphoblastic leukemia, that cancer I mentioned at the very beginning of the talk, that's called PH-like ALL. It's called this because it looks sort of like it has what's called the Philadelphia chromosome, which was actually discovered across the street a number of years ago. And a scientist in Oregon, Drucker, actually in Philadelphia chromosome-positive leukemia showed that there was something called a tyrosine kinase inhibitor, a certain drug that was very effective in that. And there are other types of ALL that may also respond to that group of drugs. And there's a number of labs at CHOP that have done research studying targeted therapies for this type of ALL, and Sarah Tajian is currently running a very complicated clinical trial that takes patients' blasts when they're first diagnosed, so leukemia cells, does a lot of complicated genetic tests to it, sees if you're somebody who's more likely to benefit from one of two newer therapies, and if you are, offer in the context of a clinical trial patients and families the ability to try one of these drugs to see if it helps. We don't know if these will actually work in this type of leukemia. In the lab, they work very well. In theory, they should work very well, but we still need to learn about this. This is just one example of clinical trials that are ongoing at CHOP, developed based on research that happened at CHOP. Another example is in a tumor called neuroblastoma, targeting something called ALK. Scientists at CHOP, including Yael Mosé and John Maris, it's probably been 15 years ago now, it showed that a certain percentage of children with neuroblastoma have a change in the ALK gene, and this may be targetable by certain drugs. And they've done a lot of studies in the lab and then some clinical trials that show it may be effective. And we have trials that are ongoing targeting ALK and neuroblastoma and relapse, and Rochelle Bagatelle, who's the head of the Neuroblastoma Disease Committee for COG, is helping run an international trial where they're looking at targeting ALK in certain patients with neuroblastoma who have certain lesions in ALK, changes in that protein gene. Another type of cancer trial that's ongoing at CHOP that's being run by Yael Mosé and is getting to be more common in cancer medicine is what we call a precision medicine basket trial. So as we kind of mentioned before, a lot of clinical trials take a long time to get from start to finish. You have an idea, you test in the lab, you want to start trying a new drug in a hard-to-treat cancer, and you open a clinical trial just to treat that rare group of patients with that one drug. A lot of times, though, that's a lot of work to get open something that's going to only affect a few children. The concept that's being done now is basically to take a disease like neuroblastoma in children who have hard-to-treat neuroblastoma and test the genetics of that neuroblastoma, knowing that some children will have one genetic change versus other children will have another genetic change in the neuroblastoma cells versus other children will have a third. And instead of having 3 different clinical trials to test 3 new drugs, in one kind of basket trial, you allow all multiple types of kids with different types of neuroblastoma to be enrolled to get access to a number of different drugs. And this is stuff that was spearheaded in the lab here at CHOP and then led to the Nepenthe trial that's open right now. The list of targeted therapies being studied in the lab or studied in the clinic continues to go on and on at CHOP. There are studies in brain tumors. There's one looking at a Czech and WEE1 inhibitor in patients with neuroblastoma and also brain tumors. Rheingold is running a trial based on some studies that went out of performed in labs here, looking at another class of agents. Leukemia. There are studies in brain tumors, including some of our investigators like Mike Fisher. And then there are also other small molecule inhibitors, drugs to treat other rare types of childhood cancers. Every day there's new trials that are opening based on some of the exciting work that's coming out of CHOP. Another area of clinical trials that's currently happening more frequently at CHOP is in the area of bone marrow transplant. So bone marrow transplant as a concept has been around since the 1970s. And the idea is if you have a disease that's of your blood cells, you can give chemotherapy or other medicines to basically wipe out your blood-producing cells and replace them with somebody else's to give you healthy, and that could be immune cells that fight infection, or red blood cells, for example, if somebody has sickle cell disease, or if you have leukemia, a way to wipe out cancer cells. The problem with bone marrow transplant historically was, do you have a match? To get a bone marrow transplant and it not be too unsafe, you have to make it so the person who's getting the transplant won't attack the donor cells. And you also have to make it so the donor cells don't attack the patient, something called graft versus host disease. And it used to be that if you were white, there was probably about a 90% chance in the US you'd have a match, because you may have heard of blood drives and bone marrow transplant drives. However, for a lot of our ethnic and minority groups, the odds of having a match were far less, just because the population frequency was lower. Nowadays, we have a way of taking bone marrow cells from a donor and manipulating them in a lab so they won't attack the person they're donating to. So we often do what's called a haploidentical transplant, which means we use a mom or a dad as the donor for a transplant. So we change the bone marrow cells so they won't attack the patient. And because of this, 99% of the time, if we think a transplant is needed for either a cancer or another blood disease, we can find an identifiable donor, either by somebody in the general population, what we should call an unrelated donor, or by using a haploidentical transplant. The survival for children who are getting haploidentical transplant nowadays, basically meaning manipulating the cells in a machine outside of a patient, has gotten to be the same as getting a transplant the old-fashioned way, using somebody who's a match and a relative stranger, and the field has changed remarkably. And Nancy Bunnan and Tim Olson and some others at CHOP are running a number of trials that are looking at different ways of manipulating bone marrow transplant outside of the patient to improve cure rates for a lot of different diseases. One of the other big areas that's been a breakthrough that has had a lot of popular press has been in cell therapy, and one of those is CAR T cells. So CAR T cells is basically taking part of the immune system of a patient, taking it out of the patient, and genetically engineering it so it attacks cancer cells. The first type of CAR T cells that was ever studied in children was done here. That was in that same type of leukemia I mentioned before, ALL, targeting CD19. And it became the first FDA-approved gene therapy, Tisagen Lec-Lucel. And then currently, this field is exploding, that there are CAR T cells for a number of different leukemias and lymphomas and other cancers as well, based on CAR T cells that were developed at CHOP. We're currently developing new ones every day in partnership with Penn, and also work that's being done here, plus CAR T cells developed at other centers that we're opening as clinical trials as well at CHOP to treat a number of diseases. There have been a lot of big breakthroughs in the CAR T cell space in what we call liquid tumors, leukemia and lymphomas, and we're now just starting to test them in some solid tumors, and hopefully we'll see the same type of benefit. Other types of cell therapy, which is basically taking cells from somebody and genetically reprogramming them, we're using those to attack viruses. So in bone marrow transplant in particular, one of the complications or side effects are some normal cold and flu type bugs can be life-threatening after bone marrow transplant. We can actually use the immune cells of either donors that are related to a patient or unrelated donors that are designed to attack different infections and use them as a therapy for some of those viruses. There's also a number of exciting gene therapies that are ongoing, and new ones are being developed every day. Children with a lot of diseases that don't have cancer, like sickle cell anemia, thalassemia, hemophilia, are now being cured with modern-day gene therapy approaches, a lot of which are run through our cancer center, because part of gene therapy, for some diseases at least, requires getting chemotherapy as part of the gene therapy, and that's something that the cancer doctors do best. And there are a number of physicians and scientists at CHOP, some listed here, Steve Grupp, who runs our CAR T-cell program, Nofia, Shannon Maud, Richard Affleck, Timothy Olson, Nancy Bunnan, myself, and others. We're working on different cell and gene therapy approaches. We don't just have clinical trials to try to cure cancer. We also have trials to try to help patients and families either through psychosocial means or through supportive care. One of the big side effects of cancer therapy is infection. Another big side effect is getting sick, nausea, vomiting as a consequence. So testing new ways to treat those. And some of those trials are with medicines, and some of those are with things like yoga or acupuncture to try to help the physical and mental well-being of patients who are going through something so difficult as treatment for cancer, which sometimes can take many years from start to finish to cure somebody. And then finally, some of our therapies have a side effect where they can affect fertility. And Jill Ginsburg has a program at CHOP for patients who are getting therapies that may impact fertility, either boys or girls, preserving either testicular tissue or ovarian tissue or sperm banking or egg freezing to make it so when those children grow up, they may have the ability to still have children in the future. I'd just like to conclude by saying it really takes a team to do clinical trials and clinical research at a place the size of CHOP. It's not just doctors and scientists, it's nurses, it's nurse practitioners, it's social workers, it's psychologists, therapists, child life specialists, our nutritionists, our pharmacists, our spiritual advisors, our administrators, and that's just the clinical side. On the research side, we also have clinical research assistants. These are people who, when you're on a clinical trial, you want to make sure you capture all the data, you do everything by the book, so you don't hurt anybody. And at the end of the day, when the trial's over, you want to make sure you understand the results. People in the lab who do research, regulatory specialists who help with some of those submissions of the complicated paperwork to the FDA or the IRB that Megan was mentioning about, people who work on budgets and contracts, our computer and IT people, the people who helped set up this meeting that we're having today, because Megan and I would never have been able to do it, people involved in compliance and monitoring. We want to make sure that we're doing it the right and the ethical way, and we have people who check on us. Their job is to come in and double check what we're doing to make sure that we're doing everything by the book. And then we have, as we mentioned before, ethics and review boards. And I just like to conclude by saying that we know that it can be very overwhelming. When families have a child who's diagnosed with cancer, that you hear that and you hear nothing else. And then you have physicians coming in there and offering lots of different trials and consents, and we understand. For example, a child who's diagnosed with leukemia, the first night, we usually have a young doctor come in there and talk about they want to do a bone marrow and a spinal tap to see if they can diagnose the leukemia. Then they talk about consenting for genetic testing to understand the type of leukemia. Then there's consents for blood transfusion. Then there's consent to have a line that's put in so you can give the medicines. Then we say we want to understand more about your cancer. So we start getting into research and we ask if we can collect extra blood or marrow or tissues that we can study at CHOP. We also want to send some of that to the Children's Oncology Group to learn and understand more too. Then we come to you with a 50-page document that talks about the 20 different drugs we want to give you over the next month. And then we may also talk about how there's a new blood thinner to prevent blood clots, consent for a lot of our behavioral studies I just mentioned, consent for the new drug to prevent infections, and then a consent to understand the long-term outcomes of having childhood cancer. It's extremely overwhelming, and it's important that you realize it's an ongoing process. Always ask questions, and you can always change your mind. You can decide to enroll in a clinical trial and then say, this is not for me, change your mind at any time and come off of it. Because ultimately, our goal is to cure more children, and if we knew all the answers, we wouldn't need to do clinical trials in them. Thank you. You might have questions, I don't know.
Yeah.
15 minutes. This is where we're getting back to how we need those IT people.
Yes, exactly.
If anybody has questions, they may be able to type them into the chat, and we may be able to answer them. I've used may twice there because I'm not 100% work properly. And it's okay if nobody has questions. All right, with that, thank you very much for tuning in.
Thanks.